virus that's been going around killing people. And no, I'm not talking about coronavirus. This virus has been around for much longer, and it infects approximately 37.7 million people worldwide. In Sub-Saharan Africa, we're responsible for a staggering 2/3 of this figure. This is the human immunodeficiency virus, or HIV. And although HIV has been around for longer than I've been alive, there's still no cure for it. When HIV enters the body, it wants everybody's attention, and it instructs the new cells it infects to make more copies of itself. These copies then go on and infect other healthy cells, and thus spread infection throughout the body. A person that is infected with HIV takes ARVs, which have now been reduced to just 1 pill a day. However, when a person becomes infected with HIV, the virus lies asleep in some of these cells. So this means that the cell is infected with the virus, but it is not producing new copies of itself, and thus it cannot be targeted by the ARVs. What's worse is that these cells cluster together to form what we call the HIV latent reservoir. The HIV latent reservoir thus is a major barrier for finding a cure for HIV. Also, we don't even know how these reservoirs are formed. What we do know is that HIV is made up of 9 genes. And no, I'm not talking about these genes that I had to jump to get into this morning. I'm talking about DNA, which makes you and I human, but also unique in our own special way. HIV uses a very special gene called TAT, and TAT plays a very important role in HIV replication. Changes in TAT have been shown to either increase the rate of HIV replication or decrease the rate of HIV replication. When TAT is not there, HIV replication does not occur very efficiently. However, TAT does not perform this very important function alone, and it has help from another protein called P-TEFb. Together, TAT and P-TEFb work together in enhancing the rate of HIV replication. What my study investigates is how changes in TAT affect its ability to work with P-TEFb. Also, we want to know how to wake up the sleepy virus so that it can begin to replicate again and thus be targeted by ARVs, thus edging us closer to finding a cure for HIV. Thank you.